Good morning, everyone. Good morning, everyone. Ann taught me that. Right. Good morning, everyone. Thank you. It is so great to be here in person with a full house. And I got to tell you, Walter, I think you have brought the heavens out and brought people together again.
I think it was Shirley.
Well, it's a real honor for me to introduce this year's C. Miller Fisher visiting professor. And I'm actually so moved by this occasion that I had to write down what I wanted to say, which for those of you who know me, that's a rare event. As many of you know, Walter grew up in Brooklyn and then went to medical school and internal medicine training in Chicago before coming here for his neurology residency. Walter graduated in 1985 from a residency here 10 years before I graduated. And he quickly earned recognition and escalating leadership roles in the department under Ann, led our efforts in stroke, neurocritical care, and ultimately became the vice chair of the department before leaving MGH after almost 30 years to become first the deputy director and now the director of the NINDS. And you can read his biography in the pamphlet. You can look at his CV online. I don't have to tell you all the amazing things that Walter has accomplished. But for me personally, it's an incredible honor and privilege to be introducing Walter for a talk in a place, in a room where he doesn't need any introduction because everybody knows who Walter is at the Mass General Hospital, because he embodies everything that the Mass General Hospital has offered to countless students, residents, fellows, faculty. He's a master clinician. He's a renowned scientist. He's a program builder, and he's a convener of talent who gets diverse input, lays out a vision, and gets the job done. He has transformed the NINDS into a modern, innovative clinical translational engine, and he's attracted great people who work together really for the common good. It's a remarkable thing that Walter has done. So Walter paved the way for my entire career. It's hard to figure out. He sort of looks like my father, but he's only 10 years older than me, so that's not sure how that math works. But he created roles and spaces for me to succeed, laid the groundwork for me to follow in his footsteps, but at the same time really encouraged me to seek my passion, which was to build digitally enabled healthcare delivery models. So he had this remarkable way of really listening to what you were interested in and helping you figure out how to make that happen for yourself. And I think many of us in this room probably feel the same way, that Walter really helped create the careers that we now inhabit. I often talk about the House of Fisher. Dr. C. Miller Fisher's influence can be found in generations of neurologists that he trained. And I would say in that regard, Walter is in the royal bloodline. He's the Prince of Wales in succession to the throne of Fisher. He is stroke royalty. So I tried to summarize, there's the 10 rules of Fisher that Luke Kaplan summarized. So these are the 10 things that I've learned from Walter. Number one, be your authentic self. It's the best path to success, and besides, everyone else has taken. Number two, take a lively interest in your patients, your colleagues, and strangers. Number three, invest in young people. Number four, be kind and listen carefully to everyone, because everyone deserves to be heard. Measure twice and cut once. Always think through a problem or a diagnosis thoroughly from all angles before you make an irrevocable decision. Pay it forward. Give someone else an opportunity to grow into the chance of a lifetime. Every request for help is an opportunity to mentor and to fill the world with the kind of people you want to work with and who will do good things. Don't assume you know why someone else made a choice you wouldn't have made. Show them the courtesy by asking them to explain their reasoning. Number 9, once you've attained mastery in something, start anew. Be a lifelong learner. And the last lesson, which I'm sad to say I've learned from Walter, is despite all the evidence to the contrary, there is life after MGH. We have a couple of things for you, Walter. We have the typical plaque and coin here, but I also, as you know, I have a tradition of writing limericks for our graduating fellows. And Walter never got one because he graduated a long time before I did, but I decided it's time for your limerick. So here it is.
And did you write?
Yeah. Anne started this tradition a long time ago, and I inherited it in a smaller way. To understand the patient, we must put aside ourselves and filter through their symptoms as with books upon the shelves. The patient's always doing the best that he can do. So we as faithful servants to our oaths we must hold true. Walter came to Boston from his Brooklyn stomping grounds and stunned us with his knowledge as he led us all through rounds. He embodies Fisher more than anyone I know. Through listening and questioning as doctors, we best grow. But even more than knowledge is humanity and care. Make us all feel special and eliminate despair. With Walter at the helm, we know the ship will stay afloat because NIH is now MGH, just in a bigger boat. That's right. Yeah, okay. All right. That's you. Girls.
Okay, well, that was pretty amazing. I don't think any of that was true. But I'll take it, at this age, you're not going to turn down any kind of praise. I mean, the great thing about MGH is you don't live on praise, you live on, trying to do better things for our patients and trying to understand how the brain works and then try and, convert that knowledge into things that are going to really help people. And the people we're trying to help, you talk to them, you talk to their families, and, for sure, just what kind of tragic situations they've been dealt. And so it's just a tremendous motivating factor that I think you learn here that, it changes your life. And I think it makes your life much more fulfilling going forward. And that certainly has been the case for me. So I could basically talk for hours without any slides, but I figured maybe the better thing to do is to, not reminisce too much about the path, although, that could do that for a long, long time. I got to thank, all the people here who got me, made me who I am. David Corey took me into the lab. I kind of screwed up a lot of time, but it all worked out in the end. Joe Martin, Joe got me into Huntington's disease on an elevator. I don't know if you remember this, but we got on an elevator to Kennedy 9 for something, and he said, "What are you going to do after residency?" "I'm going to go into the lab with David." And he said, "What do you do clinically?" I said, "Well, I'm thinking of this, thinking of that." He said, "Well, I thought you were going to do Huntington's with me." I said, "Okay." That was what a great experience that was, being able to do genetic testing for the first time, working with a great group that got the gene. And talk about Sheila and Anne. Wow, it was fantastic work with you. Okay, before I get choked up, let's get to the slides. So what I'm going to talk about is all opinion. It has nothing to do with my position in the government. So please do not take this as any kind of policy. It's just my opinions. And the thing I wanted to mention first is that I think that, where we are now, I think we're entering what I would call the golden age of neuroscience. And this picture is the people around the table around the golden age of physics. And so if you look at what happened to physics in those early 1900s, it just totally exploded. And I think we're on course for that in neuroscience. The trick is going to be, as we'll see, is to convert that explosion into treatments for people. And those are two separate things. They're related, but they're two different programs. Now, just to start off, again, I could talk for hours about stories about Dr. Fisher. Our offices were on the Kennedy 9 with Anne for many years, and he spent hours with me, hours and hours, always at night, very late into the night. But it was a tremendous experience being able to be next to this person. And so this is the reason I put these three things up. There's a picture of him, of course, and it's got that quizzical look. And he would, sit here and he would always have questions for you. He was the most curious person you have ever seen in your entire life. It was almost pathological curiosity. He was just so curious. But So as Lee mentioned, I don't know if everybody knows this, but I had given up on neurology as a medical student because I felt that we'd never get to any significant treatments. So I went internal medicine. And then I was wondering on what to do. You know, I was going with the cardiology or endocrine, two things. I was thinking about, and I had a patient that had an eye movement disorder, and I went to the library. Like nobody does that anymore. In those days, you went to the library, and I came upon this paper. It was just called Some Neuro-Ophthalmologic Observations. And I started reading the paper, and I got completely hooked that this was, you could see that this was somebody who could look at a human being like, you know, a cardiologist looks at an EKG, but the level of understanding was completely different. That this guy was on a completely different level than anything I'd ever seen before. And he was a scientist, and the experiment was the patient in front of him. But he had this ability to observe, to write down his observations. He had long notes. People remember those days of just his note from when he went to see a patient. It wasn't like HPI and history. It was like this running dialogue of observations of what the patient said verbatim, and then little snippets of exam. And it made very little sense to anybody else. And we'll get back to that. So anyway, so this actually changed my career. When I read this paper, I thought this was amazing. I started reading all his other papers, and I decided I'm going to go back. I'm going to do neurology. I'm going to go to the Mass General. And everybody at that time said, don't go to the Mass General. Lots of people, our friends, Joe, said the Mass General is finished. That they got this guy from Canada, and amount to anything, so you should come, you know, here. And I said, no, I really want to go because I want to work with this guy Fisher. And so I came and as a medical resident, and I came to grand rounds, you know, as soon as I got here, because Fisher was giving grand rounds in July. It was gave grand rounds in July. And up in the top, there were bicycle seats. I don't think they're there anymore. Right? No. But anyway, so I was sitting on the bicycle seats. Fisher comes in, and then there's rambling discourse, which made absolutely no sense to a medical resident. And I just sat in the back saying, oh my God, I'm too late. The guy's too old. He's not going to be able to. But of course, that was not true. That was his way of dealing with his observations, explaining them to people, and letting you make your conclusion. He didn't tell you what to think. So one of the chief residents who was before me said, I never learned anything from Fisher. I learned a ton from Dr. Adams. He told me all this stuff, the encyclopedia. I didn't learn anything from Dr. Fisher. And the truth of the matter is, he didn't tell you things like Adams did. He just made you think and taught you how to think, taught you how to do the observations, and then pushed you to make conclusions, which then he would trash. You forgot, you mentioned 3 things about the headache, but what about the other 51? I wrote this article, 54 things about headache. But so we say it facetiously, but that had huge impact on me. So when I came, I mentioned the grand rounds. The next thing I did, because of that paper, is I joined Shirley. I don't know if she remembers this, but as a medical resident, you had a consult period, and I signed up to work with Shirley because I really wanted to understand eye movements, because this paper had such an impact on me. I learned a ton in working with Shirley in the clinic. And then that was just the beginning of meeting a whole series of people while I was here who had that kind of Fisherian view of the patient being, the thing that you were going to focus on, you were going to learn from. And then the question was, how does science actually help that process? And that's what I've been doing since, due to, Anne and Joe and other people who are here. And what Fisher said, which has stuck with me, is that whatever you're doing, do it as if it's the most important thing in the world. So for people who know him, I'll just throw one example of this. So we would have rounds with Fisher on the Baker building, and the neurosurgeons would rotate. And we went in one day and Dr. Fisher said, well, what kind of interesting cases came in overnight? And the neurosurgeon said, well, we really didn't get anything interesting. We just got a back. That was like, uh-oh, here it comes. So Fisher said, well, that's interesting. I've never seen a case that's not interesting. 2 hours later, this neurosurgeon, like, why did I say that? But I still remember, he talked about the back pain. It's like, well, why do they have the back pain? Well, because there's pressure on the nerves. Well, when you get pressure on the nerve, you usually get numbness. You don't usually get pain, right? So he was always thinking, how do you fit the anatomy, the physiology with what the patient is feeling? And so he would say, we went on rounds. He saw a case, a patient who was, comatose, and you could spend hours on him with the patient, and he would say, you should spend your career trying to understand this case. So that was kind of the thing, that whatever you're doing, if you put everything into it, something really good is going to come out of it, but you got to put everything into it. And so I try to do that no matter what it is that comes my way. I always treat it as if it's the most important thing in the world. And that's been a lot of fun because you kind of get involved in things which are really exciting. And NINDS, the great thing is that, you have the whole of neuroscience that you're dealing with. So if you have ADHD for neuroscience and neurology like I do, it's great because here, I had to do stroke. I had to get a grant in stroke. We had great opportunities, but then it would be like, well, this Parkinson's thing is kind of interesting, but no, you can't do Parkinson's. You have to do stroke. But at NINDS, you see everything. So it's really been a fantastic experience going down there. And I want to talk a little bit now about what I think we can do together going forward. Okay. So the point here is that the reason why I think we're hitting this period where things can really explode is that three things are coming together. There was this explosion in genomics, which Joe moved into very quickly, got us the genetics behind different diseases, but then developed incredible knowledge about, the cellular neurobiology using these genomic tools to understand the molecular basis of a lot of, say, synaptic transmission. And so molecular neurobiology really exploded in my career. But the black box was our inability to kind of get that kind of detailed data about neural circuits. So if you see a patient in front of you, they have a amyloid in their brain, they have tau in the neurons, and you could say that person has dementia. And when you talk to them, you can sometimes find out they're a hell less smart than you are. So it's not actually the amyloid and the tau that's the issue, it's how that affects the circuits. And that's true about everything. That the patient's presentation to us is usually what we're seeing are changes in circuit function. And then we're, as neurologists, thinking of the neuroanatomy that's disordered or the neurophysiology that's disordered, but we're never able to actually see what's going on in those circuits. So for instance, we talk about with Steve Greenberg, white matter disease, see it easily on MRI scan, but we have no idea whether those white matter tracks are operating normally, whether they're sending dysfunctional information, whether they're not sending any information at all. So we have this ability now, which is coming from most of what I say tools out of the Brain Initiative, to actually interrogate circuits. And that brings the genomics, the molecular neurobiology, and the circuits together. And I think that neuroscience will really explode again. And the issue is, how do we bring that to help patients? And we need to, because neurologic disorders, brain disorders are, when you put them all together, the cardiovascular disease, half of that stroke, psychiatric disorders, apologies to Dr. Faber, are basically neurological disorders that no one else has been able to see in the pathology. But once you get at the circuits, they are circuit disorders, bipolar illness, schizophrenia. No question, brain circuits are not working normally, but we never had the ability to kind of see how that could happen. But now I think we have that, as we'll talk about. Substance abuse disorders. It's basically a iatrogenic circuit disorder. People take opioids, their rewiring in their brain changes, and they are stuck. The reward that they need to get, that I get from reading a paper in Nature, it's not, it doesn't satisfy. They have to have the opioid. This is a brain disorder that people get themselves into. So we need real answers to solve these problems. And the good news for us is that the country realizes that, the Congress realizes that, but the main thing is that scientists realize that. So the budget at NIH is not a top-down budget. We don't say we're going to do this much for neuroscience, that much for cancer. Most of the budget is what people write grants about. And really smart people get the grants. And it turned out that there's been an explosion in neuroscience. So the amount of money going to neuroscience dwarfs everything else. Now, COVID kind of maybe put in some changes there, but the really positive thing is that in this country, neuroscience is really a big driver in terms of the research effort. And there's multiple different institutes that are putting the money in. So if you have an area of science, you can get money from multiple different institutes. And our mission, as I said, is to understand the nervous system and then to convert that. But there's a lot that has to be done to make that happen. Some of that is what we call funding investigative initiated research, the ideas that people have, but also the infrastructure. So when I went to NINDS, there were a couple of things that look like they could be helped by infrastructure. So we put together networks for different disorders. So NeuroNext, which is a network run by Merit here, is first in human studies in different neurological disorders. EpicNet, with Maurizio runs, is a network to do first in human new pain therapeutics related to this issue of how to get non-addictive pain therapies. Stroke Network, which MGH is a big component of, is doing, I don't know, probably 15, 20 trials around the country in stroke. So that infrastructure I thought was important to have a kind of consistent year-to-year framework whereby people can come in and get their ideas in terms of trials done in a learning environment, as opposed to starting a trial, closing a trial, starting and closing trial. Very inefficient, not a learning experience there. As I mentioned, my philosophy at NIH has been to really protect these investigator-initiated research projects. So we have, when I first went, our pay line dropped to 8th percentile, and Story and I said that can't happen anymore, and we cut everything to bring it back to 14, and kind of holding a line at 14. If it looks like we're going below 14, we'll have to jettison some of these big projects and hold them off, because we need that kind of ability for people to see academia as a sustainable career, that there's that's not the ups and downs that make it hard one year to get a grant. So to try to keep that stable is really important for us. But then we also invest in training, especially with big efforts to build new training, especially in getting more diverse people into neuroscience, and also clinician scientists. So we're able to start the R25 program to fund neurologists, neurosurgeons, neuropathologists, emergency medicine, neuroradiologists, any neuro person during residency who wants to be branded as a scientist going forward, to have NIH pay time for them to do a project during the residency, and then they get 2 years of PGY level funding at some time in the future to kind of bridge that gap from going from residency into academia. David was so helpful to me. He put me on his grant to help me get started before I could get grants of my own. But that was a big gap in I saw in trying to move from the clinical space into research space. Now, the Mass General has really been a powerhouse. in many different ways. The people here are driving a lot of the research that NINDS is funding and NIH is funding. But I'd also say that the Mass General people that are trained here are now all over the place, and they're also leaders everywhere. So that I think that the things that Joe and Ann did have had really national impact. So NIH, so these numbers are, I asked my staff to do this. I'm not sure exactly how accurate they are, but there's a code, the way they enter this codes and the government, everything's coded. It's not accurate. It's very inaccurate, but it's very consistent year to year. So anyway, this is what we report out to Congress each year. So yeah, 2.7 billion to MGH over the last 5 years. And in terms of NINDS, we've awarded over 600 projects worth over 400 million in the past 5 years. So this is Mass General is a place where people rely to get good work done. My people, I also ask them when I go out to pick a couple of papers that have come out of the place I'm talking at, and so this is what they put together. There's stuff in cancer on abnormal lipid metabolism and how it could be targeted to treat glioblastoma. Work from Edith and Steve about detecting small vessel disease. Sid Cash, Lee Hochberg, looking at learned motor patterns related in human motor cortex during sleep. They have this BrainGate project with brain-computer interfaces, and they can, hook people up to machines when they can't, have any motor capability to control a computer or a prosthetic device. But they're also collecting data from the brain all the time, which is really an amazing ability to analyze things. And this was basically doing an experiment in those patients on how memories are replayed in the motor cortex during sleep. A study on dopamine, which I didn't know about, but that is a toxic product of melanin as opposed to eumelanin. And they found in this group, as Dr. Chen found, increase of the toxic form in Parkinson's neurons. And then the work that Brian Edlow is doing with looking at trying to predict recovery in intensive care unit in people who have, conditions, particularly traumatic brain injury, where it's really quite an amazing experiment, human experiment, to look at brain recovery of people who leave the intensive care unit in a vegetative state, and a year later they walk in and they say hi to the nurses and thank them for the care they got, and how the brain got from coma to vegetative, and over a year got to somebody walking back in an intensive care unit and talking again. It's just there's some secrets there we really have to understand, and then try to empower that to happen in people who don't get that far. But really some great secrets that are all around us all the time. Now, so at NINDS, our plan is to not leave any gaps, and our goal is to get therapies to people. So we want to understand how the brain works, but we're not the National Science Foundation. We're the National Institutes of Health. So the basic, but when we make these decisions to go in and test something in people, we generally fail. We more often fail than succeed. And the reason we fail is because we made assumptions along the way that, this happens because of X, and then Y happens, and if I intervene at X, I'm going to prevent Y, and it doesn't work because there were a whole bunch of other things between X and Y we had no idea about. And so it's usually we get killed because our basic knowledge of the problem is insufficient to actually get us a therapy that works. So this is so important to really work with people who are getting at these really secrets of how the nervous system works, because until we understand them, a lot of it's going to be shots in the dark trying to get things to help people. So we fund the basic neuroscience. About 65–70% of our money goes to that. About 20 to 25% goes to research on how the brain works. That's disease agnostic. The biggest part of our money goes to understanding basic mechanisms of disease. And then there's about 25% left that gets split between clinical trials and translational research, which is actually for us means therapy development. So at NINDS, we have a division of clinical research, real experts in doing clinical trials. They run all the networks, some of them I mentioned. We have a division of translational research in which we were able to hire people from pharmaceutical companies, and they have like a virtual pharmaceutical company within NINDS. They have the expertise to do drug development. We have contracts that go out to CROs that support the kind of things that are necessary to get these science discoveries to patients. And those things are things that you don't probably want to do. They are really boring. There's tedious. There's all these boxes you have to check before you get an IND from the FDA. And there's good reasons for that. But to have this set up where you can access these kind of resources to do that, we think are important. Now, the people we got to Division of Neuroscience, Division of Translational Research are all from pharmaceutical companies, and they came to us because their company shut down their neuroscience. So at NINDS, I can't tell a patient, well, we're not going to have a therapy for your disease because there's no industry that's interested in your disease. So we have, it's not as good as, Biogen putting their efforts behind this, but at least we can get some chance of people bringing their discoveries for a disease the industry's not interested in. And it's not for us to be a pharmaceutical company, it's for us to de-risk the process so that someone else will see, oh yeah, we can make money off of this, and then they take it. So we call it de-risking. And it's actually been pretty successful. Actually, it's a high bar to get in, but I'd say 50% of them fail and don't actually get to the end, which is an IND from the FDA. But then there have been a number that have, and then the big companies have picked them up. So it does seem to be working, and so people are interested in developing therapies for people. Two things to mention: it is not an easy process, and you can't do it as a hobby. It's something that you really have to hone in on, become an expert in. But there are people out there that can help you. Certainly a lot of MGH people are out in industry and they can help you too. Okay. So we have basically in our program, we have program directors who are generally assistant or associate professor level, who are in charge of every single neurologic condition or neuroscience area that you can imagine. But we only have usually one or two for any area. But they're really good people. They work really hard, and their job is to help other people, listen to them and help them succeed as best they can in the system that we have. The system is, everything that NIH does is public. It's on the website. Good luck finding it. It's all there, but it's really hard. So there's nothing like, you find out who the program director is in your area, send them an email. They'll at least tell you what website they should look at, but they can really help you. That's their job. So we're doing right now a project to try and see what we can do better to improve our fundamental neuroscience research. So this is research that's disease agnostic. A lot of effort we spend on disease X or disease Y. Congress tells us to do that. Patients tell us to do that. We thought we really need to spend some time on a strategic plan for the real basic neuroscience stuff that we do. So we're in the midst of that now. In terms of the therapy development stuff, all this slide is to make you see is that there's all these different programs, and they're all customized to a different type of translational research, whether it's gene therapy or small molecules or devices. And they're staged depending on whether you're early in your development space or you're kind of later on. And so you can move from early to late, but we have a whole bunch of things. The one thing I would mention, it's going to come later. So, and then in terms of the clinical space, we also have a number of different programs. Some of these are the networks I mentioned, but then a clinical trial, any idea that you have, it should fit in some mechanism. But they got to be customized. So you have to be able to talk to somebody to know which mechanism is really the right one for you. But the point is that there should always be a mechanism that works. You just have to, have the expertise to find it. Making a big push to try and do research that improves health equity in this country. could talk for a long time about it. Just to say one thing, we've been funding a study through George Howard at the University of Alabama, Birmingham, called the REGARD study. It looks at disparities in stroke across the country. It's been going on for 20 years. Disparities are horrible. People, you know, the death rates are like double, triple for middle-aged African Americans compared to Caucasians. And so we're at the point now where I tell people we're not doing any more of those studies. They're done. We know it's a bad problem. What now we're going to do is try and fix the problem. So no more grants to admire the problem. We want to fix the problem. So we're really pushing people to get out of their comfort zone, think about interventions that can actually help the problem going forward. So we'll see what happens. This is just kind of starting out. Richard Benson's a stroke neurologist from Columbia who's running that. Biomarkers. So I spent a lot of time in biomarkers, and I went to NIH. We looked at the biomarker portfolio. We funded a lot of grants, and none of them ever were useful for anything. So decided we're not going to do that anymore. We're going to do it differently. And what we have now is a program that looks like drug development for biomarker development. So you have a project, okay, come on in. We'll give you money to check it out, to get it started. Then we'll give you money to validate it in a really rigorous way, an analytical validation, then a clinical validation. And the hope is when this comes out, it's going to be really useful. Now, we're really interested in biomarkers to inform phase 2 trials. Because when you talk to companies about developing treatments for neurologic diseases, they will say, adios, unless you can say, here's a biomarker, and if you can affect that biomarker, it's a likelihood that this is going to work. And that's what we need to get the industry hooked on the neurologic disease. There's lots of targets out there. That's oftentimes not the problem. The problem is, how would you know if your intervention is actually hitting your target? And that's where the biomarker space, and certainly working with Gil and his group over the years for imaging, I think in stroke, that turned out to be, you know, really gold. And really influence how, you know, certainly large vessel occlusion is treated. The next thing I wanted to say, and this is, and then I don't mind if people talk about it and people criticize me, but I think in general, for many of our diseases, we're kind of hitting the top on what small molecules are going to be able to do. You give somebody something to swallow, and it goes everywhere in the body and the nervous system. To get the effect size you need is really difficult without getting off-target effects somewhere else. But the genomic therapies are precise. It's really quite amazing. And what I see in the future is therapies based on really precise targeting of particular genes and particular cell types where the other cells are completely, utterly not affected. And then you can get really big effect size on what you want to do. You want to shut down a certain neuronal group. You want to change a gene to express some other protein that then antagonizes a deleterious protein. Those are the kind of things that you can do in a dish now that I think is using these genomic techniques. And the question is getting that into the brain and doing that in the brain. But it's going to be a hard problem. But I think that these really serious neurogenetic disorders where small molecules are never going to work, and these kids have no chance of a reasonable life, this is the place to start. So these neurogenetic disorders are really kind of, even at NIH, we have all these different institutes, but a lot of these gene therapy programs are really going after neurologic disorders. And they're kids, and because there is no alternative. And so you can take big risks. And you got to be careful as you can. And there'll be bumps in the road, but I think this is going to be really kind of help us. So I've been trying to push myself to learn more and get more involved. So we have what's called an accelerated medicine partnership for gene therapy for rare disorders. And this is, again, this is disease agnostic. It's industry partners working with the FDA and NIH. We'd like to develop a platform to deliver with AAV, genes for rare disorders so that the company doesn't have to start from ground zero all the time. You build a platform, you change your gene, you plug it in, and you get, a really kind of fast pathway to test it, and then FDA approval. And this is what the FDA wants. they're really scared that there's all these different diseases and there's all these different gene therapies, and they're not coordinated. They don't know what to make of them. So they say, oh, go back to ground zero and show us X, Y, and Z. If we can do that in a platform, this could potentially be much quicker. We at NINDS set up an ultra rare gene-based therapy program, which we're going to hook into NeuroNext. So more work for Merit and her team to try to get the NeuroNext platform, which has been very effective, to try and bring in some of this genetic therapies, which I think also will, in the beginning, be mostly for these ultra rare disorders, but I think over time will expand into other disorders. I work on the Undiagnosed Disease Network, which is a really interesting network across the country that sees people, mostly kids, who have diseases that no one could diagnose. About 17% of them were found to have new genetic causes, some new diseases. 80% of the kids have a neurologic problem. So when the Common Fund project ended, NINDS kind of took this over. So we're going to try and run with this. I'm meeting with Joe about this last summer. And so again, trying to get at these new genetic disorders and match it, not so we can diagnose it as much as we can then move to, one of these bespoke gene therapies. And the most exciting thing, of course, is the somatic cell gene editing. So that's a Common Fund project that I run with Joni Rudder from NCATS. And the first 5 years, we were building tools to do gene editing, to test for off-target effects, trying to improve the safety. But now we're actually moving to try and test things to go into patients. So we have a couple of projects, and I'll bet you Ann knows that Jennifer Dudner is working on gene editing for Huntington's. So, man, we got our fingers crossed. But yeah, so of all the things, 2 of the things we're going to be doing are in the neurospace. We're only going to be doing like 5 things. So, okay. And then let's see, how are we doing for time? Okay, I'm going to just run through. I kind of talked in general, and some of these themes will come back again. But I was going to end with some of these kind of what we call targeted projects. So the preface is that, what we would like at NIH is for Congress to see the value of research and to fund the NIH fully so that we can fund all the good research that comes into us. And truth of the matter is, we cannot do that. Never been able to do that as far as I know. Every time we've looked, so we have a pay line, say 14th percentile. Anytime we've looked at what happens to people who just missed the pay line, say for instance, we get more money, we pay people above the pay line. We compare that to people who just got their money. There's no difference between the 2 groups. So we know that somebody who has a 14th percentile is no more likely to be successful than somebody who has a 20th percentile. Turns out it's about a plus or minus 5 giveaway. So we have a pay line system. We could just as easily say between 10 and 20 percentile, we'll just flip a coin. It would be equally fair. It's a complete convention. So if you don't get funded, it's not because NIH doesn't like you. It's not because you couldn't be good. It couldn't work. It's just that the way the game is played, you just didn't hit the pay line space. And that is a convention. So you can get really mad, but you got to let that die. Go in the closet, yell and scream. Do not yell and scream in front of your lab. Because then they think, oh, this is not a career for me. And I think that's been a problem. I think we're suffering from the fact that investigators who have funding, who have been working, complain, and people say, well, I don't want to be like that. So let's do a little less complaining. You complain to me, but don't let your lab people know about it. Okay. So what Francis did is we had flat years at NIH between 2004 and 2016. Francis Collins kept pushing Congress, didn't work, and all of a sudden he hit upon success story, which is I tell the Congress a story, and the Congress then says, great, let's do that. And they give you money for that story, which is not what you want, but that's what happened. Now, The good news for neuroscience is that most of those stories were about neuroscience. So we got really lucky in a kind of a bad way. But the one difference is the Brain Initiative. So the Brain Initiative did not come from NIH. The Brain Initiative came from the White House, actually. And a congressman came down and I gave him a tour and I took him, to the MR unit, and he was interested in education, how kids learn. And that got him interested in neuroscience. He said, well, what would it take for us to learn how the brain works? So I gave this big spiel about all things we could do to understand circuits of the brain and how, we're learning so much about the molecules. And I showed him the MR stuff. And he said, I never forget this. He turned around and I said, we can do this. And I said, oh, that's great. I said, I'm so impressed that you have such enthusiasm, but how do we get more people on board? And then he said to me that don't worry about that. That as long as we do something that no one's going to oppose, we can make sure it happens. And so I thought the guy was crazy. He is in jail now. I think he's staying there. Anyway, then the White House sent people down. They started asking questions, and all the IC directors would come up with their ideas. And the IC directors were not very good at this. They couldn't come to any kind of conclusion. But somebody in the White House decided we're going to do circuits, and so the president announced it. And I don't think anybody in NIH knew this. He announced it the State of the Union message. And then what happened was we put a plan together to get the money out the door, and the plan was fantastic. Really good team put a great plan together, and it's all focused on circuits. How do we understand how the circuits work? And then how do we understand how dysfunctional circuits cause disease? And how do we change those circuits so that the manifestations of disease are ameliorated? So that's really what it's all about. But it's really complicated. And the reason is because we didn't have the tools to look at circuits. So we have, you can do PCR, you can do genomics, sequences, fine, but what are the tools to look at circuits? So, I joined David's lab because I was interested in how ions cross membranes, and I had worked in artificial membranes, I worked in frog skin and cornea, and then patch clamping came, and I thought that was going to be the answer to everything. So I remember David had this chair with, contraption to do patch clamping in the OR in human beings who are alive. That was a joke. But, it's not. There are people who are, taking brain out and doing that. So there was a tool that really expanded on certainly understanding channels, but the tools for circuits really lag behind. But now they're really out there. It's really quite amazing. So instead of trying to put an electrode in the frontal cortex to look at an eye movement now, you can basically record from 1 million neurons simultaneously. And so the problem now is not that we can't get the data. The problem now is we have so much data, we don't have to understand it. But the answer is in the data. The circuit activity we can capture now. And so these rules, so I mentioned about what Sid's paper was, those kind of discoveries are now out there for the taking. Certainly in the mouse, where you can use these tools to make these great recordings, but even, what Lee and Sid are doing are in living human beings, and Sid Williams, another one. So the plan going forward is to build on these 3 areas. One is to get a cell atlas of the brain. The second one is to understand how those cells are connected to each other, and then to develop precision access to those cell types, to those circuits. So those are the 3 things, and the therapeutics come out of the precision access. Now, the people who are building the precision access, they're not that interested in therapeutics right now. What they're interested in is showing causality. Because when you have a network like this and you see all this activity, the question is, what cells are responsible for what? And if you just collect the data and correlate it with the behavior, you get a correlation. But if you can shut a nerve cell down and shut the behavior down, then you get the causation. So they want precision access to get the causation out of correlations. But the advantage for neurologists and psychiatrists is going to be precision access for therapeutics. So the brain cell census exploded heavily due to the work that was started by Evan McCasco, who's a psychiatrist now over at MIT, to develop a high throughput way of looking at single cells in the brain and their transcriptomics. And that allows you to cell type the cells in a very rapid through manner. The great thing for neurology, psychiatry, is that you can get almost the same answer from the nucleus, from the RNA nucleus, or from the chromatin pattern in the nucleus. You know, what chromatin is available, what chromatin is bound and not available to make RNA. You get very similar things out of the nucleus. And the reason why that's cool, because Josh Sain said this to me at a dinner once, that any experiment which starts when you put the brain in a blender and you turn the blender on is going to turn out really well. And the nucleus turns out very, very conserved under many different conditions. So we can now do this in human brain tissue. We don't need to take, this fresh brain out right away. This nucleus is really quite good. So you get the same kind of information out of the nucleus, but then allows you to look at the human brain, which then allows you to look at any disease you want, and you can get this kind of information on what's happening to the cell types. So are there more astrocytes? Are there less astrocytes? Are there more pyramidal neurons, less pyramidal neurons? But more importantly, it gets you at not just cell types, but cell states. So what happens in disease is the state of the cell is changing over time, and from neurodegenerative disease, it's eventually dying. Now you can actually chart that out in a human brain, because you'll have a piece of brain that in a disease that are not yet affected, but they will be affected. You'll have parts of the brain, the cells are already dead, and there's a reaction to it, and you'll have things in the middle. You can see the whole disease range, maybe in a single human brain. So many of the projects are now going after this. And then in terms of so then to get back to the precision cell access, the key thing is that this knowledge from the different cells gives you cell-specific enhancers and promoters that are really specific to that cell type. Once you find those, you can hook up any artificial gene you want that can then target just that cell type. So that's what gives you the precision cell access. So the cell census gives you keys that give you access to specific cell types. And that allows you to manipulate the biology of those cells or the activity of those cells to change circuit function. And that's really the beauty, I think, of what we're coming up with the brain energy. Now, getting access to the brain is a big deal, and there's a lot of effort now going into that. So a lot of people, MIT, Caltech folks, looking at trying to find viruses that are going to be really good at moving things into the brain. So we always have to, really keep a very strong focus on how to deal with that blood-brain barrier in a safe way. But that stuff is happening. The connectivity is also going on. And so initially the idea was we do electron microscopy of an entire mouse brain. It would take 15 years, lots of money. And I think we can pull it together. We hopefully can do something like that. But the really cool thing is that these light microscopy techniques came on, which are almost, they can't get you to EM, but they get you darn close. So expansion microscopy, where you can clear the brain, get all the lipid out so it's translucent. You can stain it with anything you want, and then you can expand it out 4 to 6 times its volume. And so now you can resolve, say, a presynaptic protein from a postsynaptic protein, and you can know, where all the synapses are for a certain cell type. So we can get almost this EM level connectivity with some of these really amazing light techniques. And they keep every couple of weeks there's a new one. There's one that came out just recently based on the scallop eye. So I'm just not going to go into it, but It's really cool. You should look at how the scallop eye works. I'll send you the paper. But it basically, just to cut to the chase, it's kind of, motivated a whole new area of microscopy, I think. Okay, so this is just an example of where, this is just a little tidbit of what's coming down the pipe. There are two papers that recently came out. This one from University of Pittsburgh, the stimulating the sensory roots in the cervical cord, and people who've had a stroke and haven't moved purposefully in years, now all of a sudden have more movement. And it's really quite amazing. I get every week, I get at least 5 people who want to get in this trial from all over the world. So this is just, the fact that this is possible is really quite extraordinary. And the question is, how does it work? So there's another paper where they did the same thing, almost the same thing in people. These are people with spinal cord injury, not stroke. And so what they did is they tried to get them to walk. They can't walk very well. They have some movement, and they do a PET scan while they're walking or right after they're walking. And what they see is a lot of activity in below the level of the lesion. So it looks like, it was a surprise. They thought they were going to find no activity. They stimulate the sensory roots and the activity goes away. And with therapy, the people start walking again. And that you would think would be amazing enough. But then the group tried to figure out what it did, so how that works. So they went into the mouse where you have these genetic tools, and they did the same experiment in the mouse. And instead of using a PET scan, they looked at CFOS activation, which is activating neurons that are very active. And what they found is, in the non-stimulated condition, there was a lot of CFOS, there was a lot of activation. They stimulate the spinal roots and the activation goes away. CFOS goes away, but then the mice start to walk again. And so then the question, well, how does that happen? This is actually opposite of what they intuitively thought was going to happen. So then they went to single cell analysis to try and figure out what cells are actually activated by this sensory stimulation, because the average condition was there was a lot less activation. And then they found a small group of cells which are positive for these genes, VSX2 and HOXA10, which are a small group in the spinal cord. And I don't think anybody knew much about them. They're connected to the motor pool. They get reticular spinal inputs. And what they found was that if you stimulate the sensory nerve roots, these cells become more active, and their connections between the sensory inputs and the upstream to downstream connection from the reticular spinal tract become strengthened. And then if they optogenetically silence those cells in a normal animal, nothing happens. They optogenetically silence those cells in an animal that's been stimulated, and the animal loses the ability to walk again. So they actually causally nail down what is actually happening. It's actually a rewiring, a strengthening of synapses between the reticular spinal tract, which has inputs from both sides of the brain and must be getting down into the spinal cord somehow. And that allows you, this is not like automatic activity, this is voluntary activity. So this is allowing voluntary activity, both the stroke case and the spinal cord case, to actually get through an alternative pathway that's not really it's seemingly not useful in a normal condition. So just a great example of how you can now dissect things out to get the causality. So what do we need to do? There's like 3, and I throw in one thing, which I think are the big challenges that I think about. One is to kind of get the right balance between the molecular neuroscience and the systems neuroscience. So one problem, I think, is that most of the people going into neuroscience now are so enamored by the system stuff that they're shying away from the molecular side. And that imbalance is not going to be healthy. So trying to do that balance. I think the future is bringing those molecular neuroscience together to see how it affects the circuits. And now you couldn't do that before. You could do LTP was like the best thing you could do, but now you can do real memory replace So bringing that molecular with the systems is important. That was unbalanced before. This is all prior to the neuroscience, they all went to molecular stuff. It's only recently that there was anything going to systems neuroscience, since except the more recent part. The other problem is data. So I mentioned a little bit before that, when I was coming out, the problem was you couldn't get the data. And now the problem is almost the opposite. Like, we have so much data, how do you deal with it? And that we got to figure out how to do. And we have to do it in a way where the data collection and data storage doesn't kill the budget for the science. That's a big problem in NIH now. But I mean, the thing I wanted to just, throw out there is that our system of dealing with science is based on the printing press. That's the technology we use. We write it down, you print it out, and send it out. That's a long time ago. Now we have the ability to do it differently, for sure. We can collect the data, we can store the data. We need people who are trained to analyze the data, and that's really where our big challenge is going to be, is in this computational space. The other problem that we have, and is that we all get enamored about our focal area, and we go for it, and then we don't pay attention to the fact that the brain is actually in the body too, and that you can get messed up on. The blood-brain barrier issue we mentioned, but there's lots of other interactions. I mean, the most amazing thing I've read, I don't know if anybody has reproduced this, is that there's actually a homunculus for the gut immune system. So you can disturb the immune system in the gut, and you can see changes in the brain, but you can also then stimulate the brain and see changes in the gut. So we are connected. One reason I went into neurology instead of internal medicine was kind of get away from the gut. Okay. And then the other area, which, just to be honest, is the big black box that's out there is the non-genetics. So we put a lot of money, a lot of knowledge, a lot of effort into the genetics of disorders, and we found out a lot. One thing we found out is that the amount that's non-genetic is gigantic, and we have no idea what that is all about. So that is the big challenge going forward, which we call the exposome. So the genome, exposome is everything except the genome. There's a lot out there. And I think the gut, the interface between the environment and the nervous system, I think, is an area of really beneficial exploration. It could be that that's where a lot of this sporadic conditions are coming from. So the gut, the nose, the mouth, these are things we neglected to look at for many years, but probably at our peril. So I can go on forever, and I would love to, but I think I just want to say I really appreciate coming back, seeing old friends, and the main thing is we got a lot of work to do, and let's get it done.
Yeah, so of course I have a question. Walter, first of all, yes, so proud of you. All these people working, and you've done so much for all of us. You're changing our country, our neuroscience. You're continuing in ways that we all hope we could, but and we're on your side. We want to help you run all the trials and get a care for all these diseases. But I just can't tell you. My heart. Yeah, it's pretty cool. And but one question I had was your thoughts on artificial intelligence, and also in this new fight. And how are we going to change this in any way?
So, ready for that, but the things that, if you look to the future, I think there are some risks, and the biggest risk, I think, is the young. So, we're people really want to use technologies or so not what we do. What we try to do is to help people who have conditions so that they have less disability and function. The problem is what I call the that's something that society really has to do. That's these brain issues for they are right down the alley having how to enhance education. Right, we learn calculus and we training cert. So all these things that you're learning about neuroscience can have huge impact on society. We've been working there. These are just now trying to get ahead of this. But this is a training that it's not going to be NIH. It's going to be in state. We're Brad Hyman on the data in the class, but they it's totally final say on medical everything. This is the one thing we start with.
Thank you. Have a celebrate.
Yeah.
You want to sit?
Okay. I'll take the mic, sir.
Are you IT? Is there any way you can turn on the